There is a pervasive modern belief that eliminating all animal products from our lives is the ultimate, uncompromising path to human health and global compassion. Yet the data tells a deeply contradictory story. An estimated 84, it merely redirects that suffering away from highly visible farm animals and pushes it onto unseen wildlife, as well as the human beings deprived of essential nutrients. This ideological drive extends far beyond the dinner plate, directly impacting medical science. Animal welfare campaigns have successfully restricted crucial primate trials and stunted the validation of lab-grown stem cell therapies. The human cost of these delays is measurable. Blocking primate research stalls gene therapies that could cure childhood blindness. Fighting against xenotransplantation, the process of transplanting animal organs into human bodies, keeps 330,000 patients languishing on organ waiting lists every year. Prioritizing the visibility of laboratory animals over the advancement of human medicine condemns hundreds of thousands of people to die from entirely curable diseases. To understand why we face these biological deficits, we have to look at the archaeological record. Exactly 2.6 million years ago, early human ancestors began using stone tools to systematically butcher and eat animals. This behavioral shift triggered what anthropologists call the expensive tissue hypothesis. Over the next 2 million years, the human brain tripled in size, becoming a massive energy sink that currently consumes 20% of our resting calories. We could only afford to power this massive brain by shrinking our digestive tract by 60%. We traded the large fermenting gut required to process rough plant matter for a shorter system optimized to absorb highly digestible, calorie-dense animal tissue. This chart visualizes the bioavailability gap, showing the stark difference between the nutrients present in food and what our bodies can actually extract. Vitamin B12, essential for nerve function, sits at absolute zero in plants. Plant-based omega-3s convert to usable DHA in the brain at a rate of less than 1%. Look at what happens when we measure iron. Our digestive system absorbs up to 35% of the heme iron found in meat. When forced to process the non-heme iron found in spinach and lentils, that absorption rate plummets to just 2 to 5%. To survive exclusively on plants today, a person must consume a heavy daily regimen of synthesized B12, DHA, iron, and zinc. A diet that requires constant pharmaceutical intervention just to stave off neurological decline and severe anemia is not a natural baseline. It is a modern industrial experiment fighting against human anatomy. That same biological reality applies to how we develop the pharmaceuticals keeping us alive. Researchers rely on approximately 100 million lab animals annually, primarily mice, operating under strict institutional welfare frameworks. While activists frequently target these highly regulated medical labs, they entirely ignore the billions of unregulated field mice crushed to death to harvest commercial grain. The medical industry faces a hard testing bottleneck. Computer models and petri dishes are excellent for early screening, but they cannot replicate whole-body pharmacokinetics, the complex way a living immune system, liver, and blood-brain barrier process a new chemical. We know exactly what happens when we skip whole animal models. In 2006, the experimental drug TGN-1412 bypassed proper animal immune testing and was given directly to 6 healthy human volunteers. Within hours, they suffered catastrophic multi-organ failure. In 1921, scientists discovered insulin by isolating it from the pancreases of dogs. Before that moment, a type 1 diabetes diagnosis in a child was an absolute death sentence. Since its discovery, insulin has saved an estimated 500 million human lives. Under a strict framework that bans all animal research, those dogs would have been spared, and 500 million people would have died. Eliminating animal testing does not remove the risk of medical science. It simply transfers that deadly risk onto the first human beings desperate enough to volunteer for clinical trials. When we remove the ideological filter and calculate the true utilitarian cost of entirely separating humanity from the animal kingdom, the math is devastating. This diagram models the stark reality of the trade-off. Attempting to save a fraction of farm and lab animals requires us to accept 7.8 billion agricultural crop deaths, wide-scale human nutritional deterioration, frozen organ transplant waiting lists, and children dying from untested genetic therapies. Throughout history, no human population has ever survived long-term without consuming animal tissue. From the Inuit hunting seals in the Arctic to the Maasai relying on cattle, our most resilient traditional cultures operated as apex predators. Even the modern nutritional science used to formulate plant-based dietary guidelines, the very data determining human absorption rates and efficiency symptoms, was mapped entirely through the animal testing that activists now demand we ban. You cannot rely on modern medical longevity, utilize life-saving vaccines, and reference established nutritional data while simultaneously trying to dismantle the exact biological research framework that built them. Human stomach acid sits at an extreme pH of 1.5. This is more acidic than the stomach of a dog. Evolution designed that hyperacidic environment for one specific reason: to kill pathogens in raw meat and break down complex animal proteins. We are the most cognitively advanced animals on Earth, and every ounce of that complexity was paid for by millions of years of consuming the animal life around us. True scientific ethics does not require us to fight against 2.6 million years of human anatomy. It requires us to accept the data, acknowledge the utilitarian math, and embrace our biological reality as obligate omnivores.